Bismillahirrahmanirrahim. Hello everyone. Our today's lecture is on purine metabolism. During this lecture, we will discuss about the structure and function and metabolism of purines. So purine metabolism, our today's point that we'll be discussing about the purine metabolism is what are purines, how many types of purines, what is the role of purine, and so on. So we will discuss about what are nucleosides, nucleotides, and their function in DNA and RNA metabolism. Remember that nucleotides, purine and pyrimidine nucleotides are the building blocks of nucleic acids, both RNA as well as DNA. Then after this, we'll be discussing about the structure and function of purines. Then how purines are synthesized, origin of different atoms in the purine ring. Remember that the purine is a 9-membered, 9-atom containing heterocyclic ring. So we'll discuss the origin of each and every atom in the purine ring. Then we'll also discussing about the essential features of purine metabolism and catabolism. And finally, we'll be discussing about the clinical aspects of purine metabolism and their deficiencies. As I point out that the purines are the building block of nucleic acids. They are nitrogen bases because they contain nitrogen atom in their ring. Look at this structure. This is, as you can see right here, it's a 9-membered ring starting from N1 from here and go right there, and this is the 9-member ring. So purine is composed of 9-member ring. It's a heterocyclic, and we have two types of purines, adenine and guanine. Both adenine and guanines are present in DNA as well as in RNA. Then the second nucleotide bases that we have in the nucleic acids are the pyrimidines. Pyrimidines are a 6-membered ring, and we have 3 types of pyrimidines: cytosine, thymine, and uracil. Cytosine is present both in DNA as well as in RNA. Thymine is present only in DNA, and uracil is present only in RNA. So that is the structural part of the nucleic acid. Nitrogenous bases, as I pointed out, there are 2 purines and 3 pyrimidines. Purines are adenine. You can look right here, the structure of adenine as well as this is a guanine, and pyrimidines are the thymine present in DNA, cytosine present in both DNA as well as in RNA, and uracil is present only in RNA. Purines are present in the form of purine and pyrimidine bases are present in the form of nucleotides, meaning that they are attached to sugar and phosphate. So they are attached to sugar and phosphate. The sugar present in nucleic acid is ribose in RNA and deoxyribose in DNA. And the phosphate is present in both. Nucleotides, as I pointed out, are composed of purine pyrimidine bases attached to sugar, ribose or deoxyribose, and finally attached to phosphate. If bases, purine pyrimidine bases, attached to sugar, then it is known as nucleoside. And if we add phosphate to sugar to nucleoside, then it becomes a nucleotide. Nucleoside and nucleotides, as I pointed out, you can see right here. Purine attached to sugar makes a nucleoside. Pyrimidine attached to sugar, ribose or deoxyribose, ribose or deoxyribose, it makes a nucleoside. And if we attach phosphate to the sugar or to the nucleoside, it makes a nucleotide. You can see right here, it's a purine nucleotide, and right is a pyrimidine nucleotide. This shows the complete structure of nucleotides, especially the purine. You can see right here, this is the adenine attached to sugar, makes a nucleoside, and if we attach phosphate, right here, phosphate to the sugar, that makes a nucleotide. One thing is to be remembered right here, that because purine and pyrimidine both contain carbon, and at the same time, the sugar which is attached to purine or pyrimidines also contains sugar atoms, carbon atoms. So therefore, in order to differentiate the carbon atoms of purine or bases and the carbon atoms of sugar, then the carbon atoms of sugar are marked with the prime sign. So it is represented as prime. You can see right here, this is the first carbon of the sugar, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and 6 prime, and 5 prime. So prime means the carbon of sugar, and without prime, normally carbon is the carbon of purine or pyrimidine bases. As far as the nomenclature of nucleotide is concerned, nitrogenous bases, adenine, guanine, and when we add or attach sugar, ribose or deoxyribose to these adenine and guanines, then they become the nucleoside. Similarly, the pyrimidines attached with the sugar also are converted to nucleoside. So in case of adenine and guanine, when the sugar is attached, then the sign SINE sign, adenosine, that is OSINE is added after the name of adenine, that is adenosine. For guanine, it will become guanosine. So the purine bases, adenine attached to sugar will make adenosine. Guanine attached to sugar is called guanosine. Both are nucleosides. But if you look at the structure of the pyrimidines, they also end with SINE. So therefore, in order to differentiate from simple purine base, pyrimidine base from nucleoside, then the name DINE is added. DINE is added after the name of the pyrimidine. That is, for cytosine, it becomes cytidine. Thymine attached to sugar becomes thymidine, and uracil attached to sugar will make a uridine. So cytidine, thymidine, and uridine are the nucleosides of pyrimidines. Then to the nucleosides, if we add a phosphate, look right here, the adenosine nucleoside, and if we add a phosphate, then it will become adenosine monophosphate. Depending upon the number of phosphates, we can have adenosine monophosphate, diphosphate, and triphosphates. So when we add phosphate to the nucleoside, then it becomes nucleotide. So for adenosine, it becomes adenosine monophosphate or AMP. For guanosine, guanosine monophosphate or GMP, cytidine monophosphate or GMP or CMP, thymidine monophosphate TMP, and uridine monophosphate UMP. So these are the nucleotides, and if they contain additional phosphate, then ADP, ATP, similarly GTP, GDP, or TDP, TTP are the nucleotides. Coming to the functions of nucleotides, what kind of function they perform in the cell? Nucleotides play key roles in many cellular processes. They are very important molecules. The most important one is that they are the activated precursors of RNA and DNA, that is nucleic acids. They are the building blocks of nucleic acids, RNA, both RNA as well as DNA. This is the first function. Then adenine nucleotides are components of three major coenzymes. That is NAD, FAD, and coenzyme A. NAD means nicotinamide adenine dinucleotide. FAD, flavin adenine dinucleotide, and coenzyme A. These coenzymes take a very essential and important part in metabolic processes. Nucleotide derivatives are also activated intermediates in biosynthetic processes. Again, the nucleotide derivatives like UDP glucose are activated intermediates in the biosynthetic processes. For example, UDP glucose and SAM, that is S-adenosyl methionine. For the synthesis of glycogen, you recall that the glycogen is composed of many molecules of glucose, and during the synthesis of glycogen, the activated form of glucose is added to the primer of the glycogen or pre-existing chain of the glycogen. And the activated form of glucose for glycogen biosynthesis is UDP glucose. That is uridine diphosphate glucose. SAM, S-adenosyl methionine, is another important nucleotide which is serving as the universal methionine donor. Universal methionine donor, adenosyl methionine. That is universal methyl group donor. Then the nucleosides serve as metabolic regulators. They control the metabolic reactions. For example, cyclic AMP and the activation of cell signaling. Cyclic AMP is considered as the second messenger. It controls many metabolic processes as well as the hormonal processes. Nucleotides again They serve as major currency of energy in the cell. ATP and GTP are the major currency of energy of the cell. Finally, many metabolic diseases have their etiology in nucleotide metabolism. These are some of the major functions of nucleotides. Coming to the biosynthesis of purine nucleotides, there are two pathways for the synthesis of purines. One is the de novo pathway. Second is the salvage pathway. De novo pathway means synthesizing the purine ring from scratch. That is, synthesizing the purine ring by adding atom by atom different atoms to make the purine ring. Salvage pathway is the one that reutilizes already synthesized purine ring or a nucleoside of purines. For example, adenosine or guanosine. So there are two pathways. The novo pathway utilizes the purine ring atom by atom, requires large amount of energy, and salvage pathway uses the recycling of the already synthesized nucleotides which are broken down. Both these pathways operate in the cytoplasm. This tells you the complete metabolic map of the nucleotide synthesis. Nucleotide biosynthesis, right here, have two pathways. De novo synthesis, as I pointed out, starting from the separate atoms or scratch, and the salvage pathways, that is recycling of the already synthesized purines or nucleosides. Both purine and pyrimidines are synthesized by de novo as well as the salvage pathway. De novo pathway of purines, purine is synthesized. The parent nucleotide, which is synthesized de novo in the cell, is IMP, inosine monophosphate, and from the inosine monophosphate, we can get the synthesis of adenosine monophosphate and guanosine monophosphate and ATP as well as the GTP. So it means the IMP or adenosine monophosphate is the parent or first nucleotide of purine which is synthesized. That leads to the synthesis of both AMP as well as the GMP. As far as the pyrimidines are concerned, the parent nucleotide which is synthesized first, or the first nucleotide synthesized during de novo synthesis of pyrimidines, is the UMP, uridine monophosphate. From this, we can get the deoxy-UMP that is converted to reduced to deoxy-TTP or thymine triphosphate. And from UMP, we can get the formation of UTP and CTP. De novo purine synthesis. De novo purine synthesis, the purine ring is synthesized by a series of reactions that add the carbon and nitrogen atoms to preformed ribose 5-phosphate. Again, during de novo purine biosynthesis, the purine ring is synthesized by a series of reactions that add carbon and nitrogen atoms to a preformed ribose 5-phosphate. Recall, the ribose 5-phosphate is synthesized as part of the HMP shunt pathway, that is hexose monophosphate pathway, hexose monophosphate shunt or pathway. Recall that HMP shunt is a bypass reactions of the glycolytic pathway. When glucose is phosphorylated to glucose 6-phosphate, then it can go to two channels. Glucose 6-phosphate can go to the fructose 6-phosphate and for the glycolytic pathway, and or glucose 6-phosphate, then by the action of enzyme, glucose 6-phosphate dehydrogenase is converted into glucono 6-phosphogluconolactone. That is the first sugar or first reaction of the HMP shunt. And during HMP shunt, we get interconversion of carbon atoms between sugars from 3, 4, 5, 6, and 7 carbon sugars. In humans, all necessary enzymes are found in the cytoplasm of the cell for the de novo synthesis of purines. This slide shows the step-by-step formation of purine ring. First step is the formation of PRPP, phosphoribosyl pyrophosphate. This is the sugar, activated form of the sugar, on which purine ring is developed. So alpha-D-ribose 5-phosphate in the presence of ATP and the enzyme PRPP synthetase is converted into phosphoribosyl pyrophosphate. Then, on phosphoribosyl pyrophosphate, the first nitrogen atom is added and donated by glutamine, which is converted into glutamate by the action of an enzyme, PRPP glutamyl amidotransferase, and we get the formation of 5-phosphoribosyl amine. 5-phosphoribosyl amine. It means we are adding the nitrogen number 9 of the ring of the purine. Then, in the second reaction, glycine molecule in the presence of ATP is added to the 5-phosphoribosyl amine by the enzyme synthetase, and the whole glycine molecule is added, and we get the formation of glycinamide ribosyl 5-phosphate. After this, one more carbon is added in the form of formyl group, donated by the N10 formyl tetrahydrofolic acid by the action of an enzyme, formyl transferase, and we get the formation of formyl glycinamide ribosyl 5-phosphate. Then formyl glycinamide ribosyl 5-phosphate. To this one more nitrogen atom is added, donated by the glutamine by the action of an enzyme synthetase, and glutamine, after donating its amino group, converted into glutamic acid, and we get the formation of formyl glycinamide ribosyl 5-phosphate. At this stage, the small ring, that is the imidazole ring, the atoms of imidazole ring are complete, and therefore a cyclization reaction takes place in the presence of ATP and the synthetase enzyme, and we get the formation of 5-amino imidazole ribosyl 5-phosphate. 5-amino imidazole ribosyl 5-phosphate. Then one more carbon atom is added, generated by carbon dioxide. By the action of enzyme carboxylase, we get the formation of 5-amino imidazole carboxylate ribosyl 5-phosphate. To this one more nitrogen atom is added, and the donor molecule is aspartate. By the action of an enzyme synthetase, and we get the formation of 5-amino imidazole 4-succinyl carboxamide ribosyl 5-phosphate. In this case, the whole aspartate was added, and then after that, the fumarate is released from this, and by the action of an enzyme, adenosine lyase. The enzyme adenosine lyase retaining the carbon and releasing the fumarate, and we get the formation of 5-amino imidazole 4-carboxamide ribosyl 5-phosphate. Then one more carbon is to be added to this one, and donor molecule is N10-formyl tetrahydrofolic acid. By the action of an enzyme, formyl transferase, and we get the formation of 5-formamino imidazole 4-carboxamide ribosyl 5-phosphate. At this stage, all the 6 atoms of the 6-member part of the purine ring are completed. The only thing left is the cyclization of the ring, and by the action of enzyme, cyclohydrolase converts this into cyclic structure, and we get the formation of inosine monophosphate. Remember, as I pointed out earlier, inosine monophosphate is the first parent nucleotide, purine nucleotide, which is synthesized de novo. From this inosine monophosphate, or IMP, then we can get the synthesis of adenosine as well as the synthesis of guanosine. And this inosine monophosphate, then in the presence of aspartate and GTP, by the action of an enzyme, adenosyl succinate synthetase, we get the formation of adenosyl succinate. Adenosyl succinate, that is, the whole molecule of aspartate is added. Then in the second reaction, fumarate is released by the action of an enzyme, adenosyl succinase, and we get the formation of AMP, or adenosine monophosphate. That completes the first nucleotide of adenine. In case of the formation of GMP, inosine is used to reduce IMP. Inosine monophosphate in the presence of NAD, which is reduced to NADH, and the inosine is oxidized by the action of an enzyme, inosine monophosphate dehydrogenase. Remember that inosine is oxidized in the presence of NAD, and NAD is reduced itself, and the inosine is oxidized into xanthosine monophosphate by the action of an enzyme, inosine monophosphate dehydrogenase. Xanthosine monophosphate, then in the presence of glutamine, that is the addition of nitrogen amino group, in the presence of ATP, and the enzyme GMP synthetase, converts xanthosine monophosphate into guanosine monophosphate. And we get the formation of glutamine is converted into glutamate. So that's how the purine bases or purine nucleotides are synthesized through the de novo synthetic processes. This chart shows you the synthesis of purine, similar to the previous one that we have discussed, but with the specific structure. The first step in the purine biosynthesis is the committed step and the rate limiting step. That is, the first step is the rate limiting, whether the protein, the purine, is to be synthesized or no more synthesis of purine is required. Second, intracellular concentrations of glutamine and PRPP also control the reaction rate. PRPP, recall that the first sugar pyrophosphate on which the purine ring is normally developed. The synthesis of purine synthesis is inhibited by AMP and GMP and IMP. These are the end products of the purine synthesis. And during the whole purine synthesis, 4 ATP molecules are required. So the purine ring that we have synthesized like outward by the process of de novo synthesis, this slide shows you the atoms present in the purine ring and the contributing molecules. You can see right here, the nitrogen number 1 is donated by aspartate, carbon number 2 and the carbon number 8 is donated by the N10 formyl tetrahydrofolic acid. Then the carbon number 6 donated by carbon dioxide. Nitrogen number 3 and nitrogen number 9 is contributed by glutamine. And you can see right here the carbon number 4, 5 and nitrogen number 7 is contributed by or by glycine. So this is very important to remember that which molecule is contributing the different atoms of purine ring. All tissues are not capable of de novo synthesis of purine nucleotides. For example, erythrocytes, neutrophils, and brain cells. They cannot synthesize. If we compare the salvage pathway with the de novo pathway of purine synthesis, you'll see that salvage pathway requires much less energy than does the de novo synthesis. Nucleotides, because they are charged molecules, they cannot enter the cells directly. Therefore, they must be converted to nucleosides, that is by the release of phosphate by cell membrane nucleosides. Nucleotides cannot enter the cells directly, and when we remove the phosphate from them, then therefore they are converted to nucleoside by the cell membrane nucleosides. Salvage pathway of the synthesis of purines. Salvage reactions are of two types. Salvage reaction by phosphoribosylation of purine bases, that is a simple purine attachment of ribose 5-phosphate ribose. That makes the nucleoside. Second salvage path reactions are phosphorylation of purine nucleotides. That is, if sugar is already attached, there are already nucleosides, and finally the phosphate is added to the nucleoside to make the nucleotide. This slide shows you the salvage reaction. By the formation of hypoxanthine. Hypoxanthine, one of the purine, in the presence of phosphoribosyl pyrophosphate, is converted into IMP, inosine monophosphate, which I pointed out is the first purine nucleotide which is synthesized, or the parent nucleotide which is synthesized. And the enzyme is hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine phosphoribosyl transferase that converts hypoxanthine into inosine monophosphate by the addition of phosphoribosyl pyrophosphate. Second, the reaction, salvage reaction by phosphoribosylation of adenine. Remember, in the previous one, we have the simple purine ring to which we added the phosphoribosyl pyrophosphate and made that one into nucleotide. In this one, of the adenine, in the presence of phosphoribosyl pyrophosphate and the enzyme adenine phosphoribosyl transferase, converts adenine into AMP or adenosine monophosphate. AMP or adenosine monophosphate. Adenosine phosphoribosyl transferase, right here shows you the conversion into AMP or adenosine monophosphate. This is the second process of the salvage reaction. Salvage reaction by phosphoribosylation of guanine. Guanine in the presence of PRPP and hypoxanthine guanine phosphoribosyl pyrophosphate. Hypoxanthine guanine phosphoribosyl transferase converts guanine into GMP. Salvage reaction, a simple phosphorylation of purine nucleosides. Remember, nucleosides are the ones in which purine molecules are attached to sugar. So guanosine nucleoside in the presence of guanosine kinase is phosphorylated and we get the formation of GMP or guanosine monophosphate. Remember, wherever in a reaction a phosphate is added to the substrate, then the enzyme is kinase. Adenosine, in the presence of adenosine kinase, and ATP is converted into AMP. In this case, what we are doing is simply we are phosphoryling the purine nucleosides. Now, conversion of nucleotide phosphate to the deoxynucleotide phosphates, because remember that the thymine deoxythymidine is the nucleotide present in the DNA. And it contains, instead of ribose sugar, it has a deoxyribose sugar. So this ribose sugar, which is present in the nucleotide diphosphate, is to be reduced to deoxyribose. And the reaction in this case, ribonucleotide reductase, ribonucleoside diphosphate, in the presence of ribonucleotide reductase, right here, you can see this one, converted into 2' deoxyribonucleoside diphosphate. So, because we are reducing the sugar, therefore, we require a reducing agent. And reducing agent is the reduced thyroidoxin. Reduced thyroidoxin reduces the ribose sugar to deoxyribose, and it itself converted to oxidized form of the thyroidoxin. Then this oxidized thyroidoxin must be reduced for the second reaction, and this reduction takes place by NADPH, nicotinamide adenine dinucleotide phosphate, reduced form, which reduces the oxidized thyroidoxin to reduced form of the thyroidoxin and itself converted into oxidized form of the NADP. The controlling mechanism of the purine nucleotide synthesis. The end product of purine biosynthesis, de novo biosynthesis, is ATP and GTP. You can see right here, ATP and GTP. Now the ATP and GTP, because both are synthesized from the parent compound IMP, therefore, if there is excess of ATP, the GTP both control the conversion from IMP to GTP, GMP or AMP and ATP or GTP. You can see right here. The excess of ATP blocks the enzyme that converts IMP to GMP so that no more GTP or GMP is required, and the excess of ATP, then GTP controls the conversion of IMP to AMP. This is the first line of control. The second line of control is from GMP and AMP that controls the first reaction, that is phosphoribosyl pyrophosphate. You can see right here, the AMP controlling the first ribose 5-phosphate and which converting into phosphoribosyl pyrophosphate by the action of enzyme 5-phosphoribosyl amine formation. Now, as I pointed out from the IMP, which is the parent compound, we get the formation of GMP as well as the AMP. As we have discussed earlier, you can see right here IMP in the presence of aspartate and GTP, it is converted into adenylosuccinate, and then the fumarate is released by adenylosuccinase enzyme, and we get the formation of AMP. Similarly, for GMP, IMP is first oxidized in the presence of an enzyme IMP dehydrogenase, and the oxidizing reagent is NAD, which is converted into NAD, reduced form of the NAD, and we get the formation of xanthosine monophosphate. Xanthosine monophosphate. Now, xanthosine monophosphate then requires amino group for the conversion of guanosine. So, glutamine is the amino group donor. By the action of enzyme GMP synthetase in the presence of ATP, in which glutamine is converted into glutamate, and ATP is converted to AMP and pyrophosphate, and we get the formation of GMP. The controlling mechanism, when we have since excess amount of GMP, no more GMP is required, then it blocks the reaction, the enzyme IMP dehydrogenase, that no more GMP synthesis is required. Don't convert IMP into GMP. Similarly, when we have excess amount of AMP, then it blocks the reaction from IMP to the synthesis of AMP, that is the adenylosuccinate synthetase enzyme. And that's how they control. Degradation of purine nucleotides. IMP is the precursor of both AMP and GMP, as we have seen in the previous slides. You can see right here, as I pointed out, IMP is the parent compound. IMP, starting with the AMP, by the action of enzyme AMP deaminase is converted into IMP, and at the same time, by another enzyme, 5' nucleotidase converts AMP into adenosine. Adenosine means we are removing the phosphate from AMP and converting into nucleoside, adenosine. Then adenosine deaminase enzyme, you can see right here, deaminates adenosine, and we get the formation of inosine. The IMP, which is formed from the deamination of AMP, is also by the action of nucleotidase is dephosphorylated and converted into inosine. Starting with the GMP, nucleotidase, 5' nucleotidase enzyme, removing the phosphate from GMP, we get the nucleoside guanosine. And then by the action of enzyme 5-phosphorylase removes the ribose from the guanosine as well as from the inosine. Right here, you can see inosine and guanosine. By 5-phosphorylase, remove the ribose sugar, convert that into guanine base, and also inosine converted into hypoxanthine. Guanine, then by reaction of an enzyme guanase, is converted into xanthine by the removal of nitrogen, ammonia, and we get the formation of xanthine. Hypoxanthine, again by the action of xanthine oxidase, which is synthesized right here, is also converted into xanthine. And finally, xanthine, in the presence of xanthine oxidase, is converted into uric acid, which is excreted in urine. So remember that the end product of purine degradation, both guanine and adenine degradation, is uric acid, which is excreted in the urine. Diseases associated with defects in purine metabolism. There are a number of diseases which are associated with the defects in the purine metabolism, like hyperuricemia, that is excess amount of uric acid in the blood, gout, Lesch-Nyhan syndrome, kidney stones, severe combined immune deficiency, severe combined immune deficiency disease. Hyperuricemia, characterized by plasma urate or uric acid level greater than 7 milligram per deciliter. Remember that the uric acid is least soluble in the blood. And the normal level of plasma levels of uric acid in females 2.4 to 6 milligram per deciliter, males somewhere from 3.4 to 7 milligram per deciliter. In certain condition literature, you may find 6 to 8 milligram per deciliter. Hyperuricemia, there are two types. Primary hyperuricemia, an innate defect in purine metabolism and or uric acid excretion. Secondary hyperuricemia due to increased availability of purines due to medication or medical conditions of or throughout the diet. So in both cases, that leads to the formation of excess uric acid and known as the hyperuricemia. Gout, a disease which is due to the defects in the purine metabolism, is caused by precipitation of sodium urate crystals in the joints, resulting in inflammation and pain. You can see right here, inflammation. See right here, inflammation, uric acid crystals in the big toe joints. You see right here, they are crystallized in the joints of the bones. Causes of gout, obviously, reduced excretion of uric acid. Remember that uric acid is the least soluble. And if they are greater than the solubility level of the uric acid, then it is the cause of the gout. The second reason is diet rich in purines, alcohol, or deficient in the dairy products. So, if we take large amount of purine in the diet, obviously that leads to the formation of excess amount of uric acid. The third point is increased purine degradation. And another one is increased phosphoribosyl pyrophosphate synthetase activity. If this activity is increased, then the greater amount of purines are synthesized, and obviously they are converted into uric acid and they result into gout. Overproduction of PRPP, phosphoribosyl pyrophosphate, leads to the increased purine synthesis, and increased purine synthesis leads to increased degradation of purines, and increased degradation leads to the increased uric acid production. Another reason for gout is decreased or partial activity of hypoxanthine guanine phosphoribosyl transferase activity. That is, deficiency of HGPRT leads to increased hypoxanthine and guanine. Deficiency of hypoxanthine guanine phosphoribosyl transferase leads to accumulation of phosphoribosyl pyrophosphate that increase purine synthesis, and increased purine synthesis leads to the increased synthesis of uric acid. Deficiency of HGPRT leads to decreased IMP, inosine monophosphate, and GMP, and that leads to the decreased inhibitors for the purine synthesis. Allopurinol, a suicide inhibitor used to treat gout. Allopurinol is the analog of the hypoxanthine, because you can see right here, the guanine and hypoxanthine both by the action of enzyme xanthine oxidase are converted into xanthine, and xanthine oxidase then is converted into xanthine oxidase converts xanthine into urate or uric acid. Allopurinol blocks the activity of xanthine oxidase. You can see right here. Xanthine oxidase activity so that guanine and hypoxanthine should not convert into xanthine and should not be converted into urate. So the treatment of gout is the use of the drug allopurinol. See right here, the condition due to gout, inflammation of the joints. You see right here, and believe me, it's really a very, very painful. Gout, accumulation of uric acid, salts in joints, as you have seen in previous slides and also in this one, you can see right here. Accumulation of uric acid, salts in joints, again, another picture. You can see right here. Look right here, right here. Gout, characterized by hyperuricemia and acute arthritic joint inflammation by deposition of uric acid crystals. Primary gout is genetic and mainly affects men over 30 years of age. Secondary gout is associated with leukemia, polycythemia, hypoxanthine guanine phosphoribosyltransferase transferase deficiency, renal insufficiency, lifestyle, for example, rich foods. Right here you can see the yellow crystals of uric acid which are deposited in the joints. Gout, kyphosis, accumulation of uric acid salts in cartilage under the skin. You can see right here, deposition. Lesch-Nyhan syndrome. Lesch-Nyhan syndrome is due to decrease or absence of hypoxanthine guanine phosphoribosyltransferase transferase that leads to overproduction of uric acid, that is hyperuricemia, with frequent uric acid lithiasis and Bazare syndrome, that is of self-mutilation, meaning finger and lip biting, which leads to mental retardation. Uric acid formation, excess free adenine in the body is converted into 2,8-dihydroxyadenine, which causes renal damage by forming crystals. 600 to 800 milligram uric acid excreted per day. Uric acid is catabolic product of purines. Excess causes hyperuricemia, in which serum urate level exceeds the solubility level, and a result, sodium urate crystals deposit in soft tissues and joints are called tophy. Excess deposition of uric acid in joints causes gouty arthritis. Treatment: the enzyme xanthine oxidase is inhibited by allopurinol, as you have seen in the previous slides, and blocks and decreases the uric acid formation. Von Gert's disease is another one. If you recall your glycogen storage diseases, Von Gert disease is also there. It is due to deficiency of glucose 6-phosphatase, which leads to elevated rate of pentose formation in HMP shunt. If excess amount of glucose 6-phosphate enters into HMP shunt, it leads to the Von Gert's disease. It leads to the increased formation of phosphoribosyl pyrophosphate. So increased formation of pentose is a good substrate for phosphorybosylate pyrophosphate synthetase, leading to increased synthesis of purines, followed by their catabolism to uric acid. Increased lactic acid competes with uric acid excretion, resulting in retention of uric acid. Lesch-Nyhan syndrome, buildup of hypoxanthine and guanine. Degradation of hypoxanthine and guanine results in increased uric acid. Excess uric acid in urine often results in orange crystals in the diaper of affected children. Severe mental leads to severe mental retardation, self-mutilation, involuntary movement, and gout. You can see the lip biting due to gout. Kidney stones. When uric acid is present in high concentration in the blood, it may precipitate as a salt in the kidneys. The salt can form stones, which can in turn cause pain, infection, and kidney damage. You can see right here the kidney stone, these stones right here, the stones present in the kidney. Summarizing the disorders of purine metabolism. Disorder is gout due to the defect in phosphorybosylate pyrophosphate synthetase and hypoxanthine guanine phosphorybosylate transferase. Right here you can see. And the result is hypouricemia. The second disorder, Lesch-Nyhan syndrome, which is due to lack of hypoxanthine guanine phosphorybosylate transferase, which leads to the formation of hyperuricemia. Severe combined immune deficiency disease, SCID, due to defect in the adenosine deaminase enzyme, leads to the high levels of deoxy-AMP in the blood, and which results into the inhibition of nucleic acid, DNA. Von Koch disease, due to defect in the glucose 6-phosphatase, that results into hyperuricemia. Adenosine deaminase deficiency expressed in cytosol of the cells. Immune lymphocytes have highest activity. Deficiency of adenosine deaminase causes accumulation of adenosine, converted to nucleotide triphosphate and deoxynucleotide triphosphate, as deoxynucleotide ribonucleotide reductase inhibited. Prevents the formation of all deoxynucleotide triphosphates that cells when the nucleotide triphosphates are not synthesized, deoxynucleotide triphosphates are not synthesized and prevent the formation of all nucleotide diphosphate, and cells cannot make the DNA. Thank you.